नमस्कार। कार्यक्रम प्रारंभ करूं उससे पूर्व मैं सादर आमंत्रित कर रही हूं आज के कार्यक्रम के अध्यक्ष श्री सज्जन कुमार तुलसियान जी से कि वे मंच पर अपना स्थान ग्रहण करें। आज के आयोजन के मुख्य वक्ता डॉक्टर शिव शंकर रॉय से मेरा सादर अनुरोध है कि आप कृपया मंच पर अपना स्थान ग्रहण करें। कार्यक्रम के कन्वीनर डॉक्टर डीपी दुआरी जी से मेरा सादर अनुरोध है कि आप कृपया मंच पर अपना स्थान ग्रहण करें। एक चेयर और चाहिए चेयर एक और चाहिए, दे दो। आज के कार्यक्रम की विशिष्ट वक्ता डॉक्टर बासुमिता चक्रवर्ती से भी मेरा अनुरोध है कि कृपया मंच पर अपना स्थान ग्रहण करें। और इस पूरे कार्यक्रम के परिकल्पक भारतीय विद्या मंदिर और भारतीय संस्कृति संसद के प्राण पुरुष डॉक्टर विट्ठलदास मुंदड़ा से मेरा सादर अनुरोध है कि आप मंच पर अपना स्थान ग्रहण करें। सर्व शब्दार्थ संधात्री सर्व ज्ञान प्रदायिनी सर्वदा सर्वतः पातु स्वर्णवर्णा सरस्वती। माँ सरस्वती के चरणों में सादर वंदन। सभागृह में विराजित समाज सचेतन लोगों का भारतीय विद्या मंदिर और भारतीय संस्कृति संसद की ओर से हार्दिक स्वागत, अभिनंदन। साइंस एंड सोसाइटी सिम्बायोसिस को लेकर जो दिसंबर 2025 में जिस कार्यक्रम का प्रारंभ किया गया था, आज का यह आयोजन उसी श्रृंखला की एक महत्वपूर्ण कड़ी है। कहा जाता है कि एक स्वस्थ महिला ही स्वस्थ परिवार और सशक्त समाज की नींव होती है। लेकिन सोचिए, अगर नींव ही कमजोर हो जाए, वह महिला जो पूरे परिवार का ध्यान रखती हो वही बीमार हो जाए, तब क्या होता है? आज हम एक ऐसे ही गंभीर विषय पर चर्चा करने यहाँ एकत्रित हुए हैं। कैंसर जो न केवल एक बीमारी है बल्कि एक सामाजिक सच्चाई है। क्या है? महिलाओं में कैंसर। कैंसर का नाम आते ही मन में डर समा जाता है। डरना कोई समाधान नहीं है, जागरूकता समाधान है। महिलाओं में बहुत तरह के कैंसर होते हैं। अभी हमारे जो विशिष्ट और मुख्य वक्ता हैं वो इस विषय पर चर्चा करेंगे ही। पर दुख की बात यह होती है कि जब महिलाओं में, अधिकांश महिलाओं में जब पता चलता है कि कैंसर है, तब तक वह स्थिति काफ़ी उन्नत अवस्था में पहुंच चुकी होती है। आज की सबसे बड़ी चुनौती है जानकारी की कमी, सामाजिक झिझक, आर्थिक बाधाएं, पारिवारिक परेशानियां आदि आदि। इतना सब कुछ होते हुए भी मैं ये कह सकती हूँ कि कोई चिंता की बात नहीं है, क्योंकि हमारे पास हमारे ऐसे दूरदर्शी अध्यक्ष जिनकी सोच जहाँ से समाज के लोगों से शुरू होती है, वो समाज को उठाने, समाज को बढ़ाने के बिना उनकी सोच का कोई अंग ही नहीं है। तो पहली बात तो हमारे पास हमारे अध्यक्ष हैं, और दूसरी बात यह है कि साइंस और टेक्नोलॉजी भी इतनी विकसित हो गई है ये चिकित्सा विज्ञान, नई स्क्रीनिंग तकनीक, एचपीवी वैक्सीन और आज जिस तरह का जागरूकता अभियान कार्यक्रम किया जा रहा है, इस तरह के कार्यक्रम यदि कई बार लगातार हों तो मैं समझती हूँ कि ये विशेषकर महिलाओं में जो कैंसर होना और उन्नत अवस्था में पकड़ा जाना वो काफ़ी हद तक कम हो सकता है। भारतीय विद्या मंदिर और भारतीय संस्कृति संसद वह संस्था है जो भारत की गौरवपूर्ण संस्कृति, साहित्य और कला से तो जन जन का परिचय करवाना चाहती ही है, साथ ही वो यह भी चाहती है कि समय समय पर साइंस और टेक्नोलॉजी के क्षेत्र में जो डेवलपमेंट्स हो रहे हैं, जो रिसर्च आ रहे हैं, उनसे आम जनता भी लाभान्वित हो। क्योंकि लोगों ने विज्ञान को एक अलग जगह रख दिया है कि विज्ञान, भाई हमारा क्या मतलब, हम साहित्य को पढ़ते हैं, विज्ञान, हम गृहिणी हैं, हमको विज्ञान से क्या मतलब। जबकि विज्ञान का हमारे जीवन में बहुत बड़ा प्रभाव है। अब हमको कोशिश ये करनी है कि विज्ञान हमारी संभावनाओं को प्रशस्त करे, हमारी समृद्धि को बढ़ाए, हमारी जानकारी को, हमारे ज्ञान के क्षेत्र को बढ़ाए। मुझे यह बताते हुए अत्यंत हर्ष की अनुभूति हो रही है कि आज के कार्यक्रम की जो अध्यक्षता कर रहे हैं, वे स्वयं, उनका ये मानना है कि हमारे देश की महिलाएं विकास करें, उनकी प्रतिभा विकसित हो, वे स्वस्थ और सुरक्षित रहें। आज कर्तव्य की प्रेरणा और दायित्व का बोध जिनकी प्रेरक शक्ति है, ऐसे कर्मठ सृजनात्मक व्यक्तित्व के धनी, इनकम टैक्स बार एसोसिएशन के अध्यक्ष, अखिल भारतीय ख्याति प्राप्त आयकर सलाहकार श्री सज्जन कुमार तुलसियान को अपने बीच पाकर हम सचमुच हर्षित हैं। मैं अनुरोध करूंगी हमारे विशिष्ट ट्रस्टी श्री राजगोपाल सुरेखा जी से, बाहर है लगता है, अच्छा, हमारे अर्थ सचिव श्री सुशांत गोपाल सुरेखा से मेरा सादर अनुरोध है कि आप आएं। आ गए। और आज के कार्यक्रम के अध्यक्ष सज्जन कुमार, राजेश दूर लेके, तुलसियान जी का अंग वस्त्र प्रदान कर उनका अभिनंदन करें। आज के मुख्य आकर्षण के केंद्र प्रधान वक्ता डॉक्टर शिव शंकर रॉय, जो न केवल उच्च कोटि के वैज्ञानिक हैं बल्कि मानवीय संवेदना से ओतप्रोत एक समाज सचेतन व्यक्तित्व हैं। आप लगातार अपने रिसर्च के माध्यम से यह प्रयास कर रहे हैं, यह खोज कर रहे हैं कि कैसे मानव मात्र को कैंसर जैसे रोग से निजात दिलाई जा सके। आपने 4 बुक्स लिखी हैं, 3 पेटेंट्स आपने फाइल किए हैं। इसके अलावा राष्ट्र निर्माण के क्षेत्र में आपने 60 से अधिक छात्रों को मार्गदर्शन प्रदान किया है। ऐसे अप्रतिम मेधा संपन्न इंडियन इंस्टीट्यूट ऑफ केमिकल बायोलॉजी के चीफ साइंटिस्ट और आज के कार्यक्रम के मुख्य वक्ता डॉक्टर शिव शंकर रॉय का हम हृदय से स्वागत, अभिनंदन करते हैं और सादर अनुरोध करते हैं संस्था के सचिव राजेश दुगड़ से कि वे आएं और अंग वस्त्र प्रदान कर उनका सम्मान करें। आज के आयोजन की विशिष्ट वक्ता डॉक्टर बासुमिता चक्रवर्ती, जिन्होंने देश विदेश में स्पेशलाइजेशन किया है, और आपको यह जानकर आश्चर्य होगा कि विदेशों में जहाँ की टेक्नोलॉजी और साइंस हमारे यहाँ से बहुत अधिक उन्नत अवस्था में है, वहीं पर इनको इतना रिकॉग्निशन मिला है। इनके जितने प्रेस्टिजियस अवार्ड्स हैं वो सारे बाहर से दिए हुए अवार्ड्स हैं। तो आप समझ सकते हैं कि ये कहाँ तक पहुँची हुई हैं। सेवा, प्यार, सहानुभूति और पेशेंट्स के फैंटेस्टिक केयर के बल पर आपने अपनी एक अनुपम दुनिया बनाई है, अनुपम पहचान बनाई है। मैं ऐसी प्रतिभा संपन्न डॉक्टर बासुमिता चक्रवर्ती का हृदय से स्वागत, अभिनंदन करती हूँ और अनुरोध करती हूँ संस्था के अर्थ सचिव सुशांत गोपाल सुरेका से कि वे आएं और बासुमिता जी का स्वागत अंग वस्त्र प्रदान कर करें। इंटरनेशनली रिनाउंड एस्ट्रोफिजिसिस्ट, एमपी बिरला प्लानिटेरियम के फॉर्मर डायरेक्टर, आज के आयोजन के कन्वीनर और हम सब के बेहद अपने डॉक्टर डीपी दुआरी जी को अपने बीच पाकर हम सचमुच आनंदित हैं, प्रफुल्लित हैं। मैं अनुरोध करूंगी श्री प्रियंकर पालीवाल जी से कि वे आएं और अंग वस्त्र प्रदान कर आपका अभिनंदन करें। मंचस्थ सभी अतिथियों का और सभागृह में उपस्थित सभी का एक बार पुनः स्वागत करते हुए मैं आप सबके बीच इस कार्यक्रम को दुआरी जी को समर्पित करूं, उसके पहले एक प्रश्न छोड़कर जाना चाहती हूं कि जब हमारे घर में एक महिला बीमार होती है तो क्या वह एक व्यक्ति बीमार होती है या पूरा घर बीमार होता है, पूरा घर उसकी चिंता में लग जाता है। कैंसर के विरुद्ध लड़ाई केवल डॉक्टरों या अस्पतालों की नहीं है, सिर्फ अस्पतालों की जिम्मेदारी नहीं है। ये कैंसर के विरुद्ध लड़ाई हमारी सोच, हमारे समर्थन और हमारे समय की जिम्मेदारी है। आइए डर को नहीं, जागरूकता को बढ़ाएं, चुप्पी को नहीं, संवाद को बढ़ाएं, लापरवाही को नहीं, जिम्मेदारी को अपनाएं। श्रद्धेय गुरुवर शास्त्री जी के मुंह से सुनी 2 पंक्तियां, उसमें किंचित बदलाव के साथ मैं आज के इस कार्यक्रम को समर्पित कर रही हूं कि कोई कैंसर तुम्हारे साहस से बड़ा नहीं। भाई जी, कोई कैंसर तुम्हारे साहस से बड़ा नहीं, वही हारा जो लड़ा नहीं। धन्यवाद। अब मैं सादर अनुरोध कर रही हूँ डॉक्टर दीपी दुआरी से कि वो आगे के कार्यक्रम को।
A very good afternoon, a very good morning to you, 10 minutes still. It's a wonderful, wonderful feeling seeing so many erudite, interested, highly educated and motivated people assembling here today to discuss something which has, as Mrs. Tara Dugar was telling, has become one of the key features of our social and also our emotional constraints. You all know what is today's lecture, the topic, cancer and women. And we have a speaker who has dedicated his academic life, his research career, and other aspects of his focus towards serving the society in trying to help elevate the critical situations of cancer-affected patients in the world, in the country, but mostly in eastern part of the country. My job has been made very easy by Mrs. Dugar because it's one of the most beautiful introduction to any program or any seminar. It's absolutely beautiful. And I'm so happy that whatever I thought that I will be speaking, she has spoken 75% of it. But you all know that cancer is not a biological disease, as was mentioned. It is a profound social phenomena. It affects families, economics, healthcare, and the psychological fabric of the community. So when we speak about cancer, as we'll be trying to do today, we must look beyond cells, tumors, diagnoses, and treatments. We must examine its impact on society and how a social hand-holding can help us in trying to, I cannot say that the whole thing will go away, but in trying to make things possible to understand, realize, and also try to go through the problem of this particular cancerous issue, cancer. I know that the word cancer itself creates a lot of, trepidation in your mind. People get afraid. There is a lot of stigma attached to it. Then helplessness, money becomes a huge problem. But my feeling is that all these things like economic impact, social burden, stigma, and psychological effects, gender in the sense women especially, can be addressed by the role of awareness and prevention. And today we are here for that, mostly awareness. What will we do with awareness? Can we reach out to only some 40, 50 people here? No. The whole concept started with Bharatiya Vidya Mandir and Bharatiya Sanskriti Sangsad deciding that apart from being a very vibrant socio-cultural organization and also skill developments in different branches, there should be a dialogue, an interaction between the society and scientists, technocrats, technologists. Because I think we all know, apart from all these very popular, slogans, India is on the verge of a big breakthrough. Breakthrough of becoming one of the most prominent nation in the whole world. And I'm not saying it just for a feel-good factor. Last year I was at United Nations attending a conference, and there I heard through my own ears the Secretary General was saying, India is now one of the top 5 countries in the world in science and technology. But somehow, our younger generation, our school kids, our college students, they were not made aware of this particular phenomena. No newspapers will have a special column for that, not even one in a year. No television channels, instead of talking about sometimes mindless political debates, will raise the issue of how the younger generation, 60% is below 35 years age in India, can contribute for our science, technology, resulting in the development, resulting in economic prowess. So in that sense, the science-society symbiosis, this concept, was nucleated with Dr. Vitthal Das Mundra some 2-3 months back. And we decided that we will do not a standard, conference kind of thing or a seminar. We'll do certain things. We'll try to nucleate ideas which will attract general, erudite, interested people in getting into a dialogue, not a monologue. Right. Because it is through this dialogue, though we are reaching today here 60 people maybe, but through each of them, we can reach a much, much larger section of the society when they will be talking about this, speaking about this, expressing their views, expressing their ideas on their own time, in their own field, in their own environment. So each of you today will be a messenger. And the messengers will be given the information by none other than our speaker. I think I don't have to talk about him because his very brief biodata has been given in the invitation, the e-invitation card at least. And I can only say that he is one of the rarest of researchers, scientists, who not only gets immersed in his research activities, looking at the computer screen or trying to tell his students what to do and what not to do, simulations, actual data sampling, data analysis, but he also has this focus in every part of his activities, how it can benefit the society. That is one of the biggest aspect of a scientist. Because we all talk about taxpayers' money, and scientists are really benefited out of that funding, that whatever funding or policy the government has. But ultimately, they have to look back and try to think how much I will return in comparison to how much I have received from the society. And Dr. Roy is one of the best example. Not just he's sitting here and I'm telling it, but I have followed his work through other friends who have told me about his research work and approach. So today he will be one of the best initiator, a nucleator of ideas in all of you, in dialogues in all of you, in thoughts of emotional approach towards science, towards cancer, cancer research, future, and everything. So that you can go out into the society and start propagating this feeling, not the information only, the feeling. We should be accountable to science and society. Not that we should learn science and deal with society. Accountable. Each of you are a successful person. But believe me, your success, that was responsible for the society, and the society made that success for you. Without society, you could not have been successful. So, time has come that not only today we'll discuss about cervical cancer and breast cancer and ovarian cancer, how Maharashtra, West Bengal, Madhya Pradesh is the leading state in the country in women's cancer, and how to mitigate that or how to address that. We should also try to think how in future each one of you should come forward on your own and advise us, tell us what should we do for the society to get benefited on discussions of this sort of science and technology oriented subjects. Please, let us not only make instruments, make in India, let us make newer ideas, newer thoughts, newer innovations, make in India. And you can be those persons who will be doing that feeling, that particular function. I express my ultimate gratitude to Dr. Vitthal Das Mundhra. He being a tremendously achieved, highly acclaimed academician, is also one of the captains of the industry. Most of the time when he said industry, we think that, okay, manufacturing things and selling things and buying things and so on and so forth. But his notion of experimentation in newer technologies. And just to give you two ideas. And apart from we seeing the name of Simplex Infrastructures last 20 years for East-West Metro and blah, 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 he was the first person who was approached by Government of India and ISRO to build the launching pad at Sriharikota Space, Satish Dhawan Space Center. How many of us know that? And with his own wishes, do you know, the only building in Kolkata and around which has not a single brick in the construction, a 6-story house, it's a total, complete concrete house. Shakespeare Sharani is Simplex House, which is a unique phenomenon. So, in that sense, I think he is the right person with his ideas of vision, ideas of innovation, ideas of incubation, and ideas of giving back to society to start off this process. And today, we are at the second program of this. The first one happened in December, and this will be a tremendous input to all our resolve to go out, reach out, and talk to the society. Thank you. Sir, Professor Roy. I think you have to get down the chairs because on the screen should be, you know.
So good afternoon. Just crossed 12 o'clock. So, so first of all, I'm honored here to be invited to give a lecture on the cancer, especially women-centric cancer. And I am really thankful to Dr. Mundra for inviting me to give a lecture in this August gathering. I also thank Dr. Duwari and all the organizers, especially Dr. Paliwal and others, for inviting me in such lecture series. I attended the first one in last month, in December, and it was so amazing. I thought I would come again, and they invited me. So it is my pleasure and honor to be here and to discuss about some of the research interest of ours, which is going on in CSIR, Indian Institute of Chemical Biology, is a national laboratory under the Council of Scientific and Industrial Research located in Jadavpur. So, so I thought I'd give my research interest here, but as Dr. Mundra suggested that it is a mixed gathering, and so please introduce the topic and in some details about the basics, so that the most of the people, are interested in learning this thing or understand this thing. So I actually kept my topic in such a way that in the first half I'll give some sort of brief introduction about the subject and the promising R&D, research and development in our country or abroad, and promising technology which is being, which will help in health sector, and also at the last some of our key important ongoing research activities at our laboratory. So there are two aspects. First is, basic aspects about the cancer, especially the women-centric cancer, and the second part, little bit of our own research interest at our laboratory. So first slide, please. Next slide. Okay, so to begin, for the beginners, what is cancer? Cancer is a group of disease. It's not a single disease. Okay, so it's a group of disease, and how it is formed? It is formed out of, it is actually abnormal cell division. And normally if you can see in the top, that is a normal cell division, which is, the abnormal, it is some cells are dying due to apoptosis. But if some genetic changes, or it is called mutations, coming in a cell, and there are multiple mutations accumulate, then the cells are not behaving properly. They are abnormally growing and causing the tumors. So there are many factors which are associated with this abnormal behavior. So basically, researchers are going on how this behavior is changed in the tumors, but mostly many of the mutations have been understood, and these mutations occurring due to different factors, and the cells are proliferating. Next. So if you go back to the history of cancer, so in the right side you can see there are almost 3,000 years ago, BC, there is some sort of evidence that there is some papyrus. They showed that there is some sort of malignancy or tumor in breast, and the cancer, the name came from the carcinoma, it is means a crab, and it is showing that crab means cancer as a carcinoma. So next slide. So there are several stages of the cancer. So there is no pointer, so I can go here. So there are different stages of cancer. In the first,
you don't need mic. You can speak. Audience is very small.
Okay.
So you can go there and do that.
Fine. So It's okay. I can give some. So in the first stage, you can see that in the diagram, these cells are epithelial cells. It's a very tiny, small group of cells, and ultimately, when there is a hyperplasia, the cell divides and becoming small tumor, and after that it goes to it is becoming bigger and bigger, and next stage, and ultimately some cells are coming into the blood circulation, and from blood circulation it goes to another area which is called metastasis, and this cancer is spreading. So this is the different stages of cancer in a very brief. And next slide. Next slide, please. So this is actually, it is called epithelial to mesenchymal transition. The cells, which is epithelial cells, you can see the structure of the cells, which is like, the epithelialness. And when the metastasis occur, that is tumor is spreading through blood circulation, the cells structure is like a mesenchymal, this type of structure, so that it can spread easily to another place, and it is growing there. This is called metastasis, and it is called epithelial to mesenchymal transition. And when it's going to another place where it resides, then again it is the opposite, is called mesenchymal to epithelial transition, and then the tumor, this is called, the tumor is metastasized. So this is metastatic potential of the cells, which is gain in the cells, and it is being metastasized. Next slide. So there are different types of cancer, as you know. This is a carcinoma. It's originate in the epithelial cells. Sarcoma in the bones and muscles. Leukemia, as you know, in the blood cancer. Myeloma is a lymph. And then lymphoma is a lymph. Myeloma is originating from the antibody-producing plasma cells. And carcinoma. So there are different types of, and there is mixed type also. So the next slide. So this is the statistics of cancer occurrence. I am just going because it is a women-centric cancer. So I am just considering the right panel. You can see there are it is in the world population. It is the Globocon 2022 data. You can see that. Among women, there are about 24% is having the breast cancer, and you can see the ovarian is about 3.4% in the worldwide. And mortality is also 15% in breast, and ovary is another 4.8%. So, and if you go to the total number of new cases, it is a 97 lakhs in worldwide, and death is 43 lakhs. So in next slide, in India, the situation is almost similar. You can see that breast cancer incidence, it is new cases is something 691,000. 6.9 lakhs, something like that, and the mortality is also very high, and it is also in the ovarian cancer also the number is very high. So next. So what are the different risk factors? You can see that risk factor is maybe genetic, maybe infection, maybe environmental factor, maybe lifestyle. So genetic means there is some mutation already there. Like in case of breast and ovarian cancer, there is a BRCA, BRCA1, BRCA2 mutation is there. There are other mutation in different other cancers also. So that is the risk factor. And also there is infection, like in cervical cancer, you can see there are human papilloma virus infection is there, and for that it may cause cancer. Environmental factor is different radiations, UV, gamma, and different type of radiations, carcinogens, and lifestyle also, the tobacco and smoking also, and different other factors. They are the risk factor. So this may cause, they may lead to cancer. Next. So this is actually the schematic diagram to show that intrinsic and extrinsic biological factors, that cause to cancer, you can see the different agents like biological agents, genetic mutations, inflammation, and chemicals, carcinogenesis, dysregulated metabolisms that leads to cancer. And when it comes to the tumor cells, that tumor cells are having the all the processes or all the characteristics that is called hallmarks. So these are the hallmarks of tumor cells, that is their replicative immortality, avoiding immune cells, metastasis and tissue evasion, evading growth suppression, tumor heterogeneity, undisrupted angiogenesis, cell death, resistance, etc., etc. So what is there? So if there is a sort of mutation or any other cause, the tumor cells become, normal cells become tumor cancerous cells. They go through some sort of hallmark. They have some characteristics, which is, it is called the hallmarks. Next. So there are some diagnostics. How we can someone diagnose is a clinician's job. They by seeing the medical history of the patient, then either medical history, then biopsy. There are different types of biopsies. The imaging, different imagings like X-ray, MRI, CT scan, ultrasound, etc., endoscopy, and other tests like genetic testing. So these days the genetic testing is very important. So these are the different aspects by which you can detect or diagnose the tumors. Next. So there are several markers available, which is how these markers are coming because of the, when there is a tumor, there is some sort of biochemical or other reactions inside the cell. So some antigens are coming, or different antigens or enzymes, mainly proteins are overexpressed or coming out of irregular metabolism into the bloodstream, and you can test it. And when you can see their level is very high, you can predict that it might due to the tumor. For example, CA125 in ovarian cancer, PSA in prostate cancer, and there are so many other markers. When it comes into the blood, it is showing that it might be having the, started the tumor progression. So next. So treatment strategy, actually there are several treatment strategies. So mainly depending upon the stage and the nature of the cancer, clinician decide whether to go for the chemo or radio, surgery, or immunotherapy, or in combination. So you all know what is chemo and radiotherapy and surgery. I'll be giving some details about the immunotherapy because it is relatively newer technology, and there are a lot of scopes for R&D, research and development, how we can be coming in a bigger way. So next is, and also hormone therapy in some diseases like breast cancer or ovarian cancer. So these are the different treatment strategies, and also some newer strategies are coming up. So I'll also discuss a little bit about it. Next. So this is actually chemotherapy. I'm not going into the details. There are different drugs available, mostly basically DNA targeting drugs like platinum drug, and also the it is actually blocking the DNA replication so that the cells cannot grow. But the problem of this type of drug is like it also kills the normal cells apart from the tumor cells. So this is a little non-specific. And there are also drugs like topoisomerase inhibitor, which is the enzymes which is helping the DNA replication. It blocks that, like doxorubicin. And also there are some drugs like Taxols, which is actually binding to the tubulin, and it blocks so that the cells cannot divide. So these are the different types of drugs basically. Now there are some targeted drugs are coming, has come. A lot of targeted drugs has come, which is actually targeting specifically some proteins, and so that the tumor cells can be killed specially. So these are the basically different chemotherapeutic drugs. And next. So there is a radiation therapy. I am not, going. There are two types of radiation. One is the external beam and also the internal. So depending upon the structure, this is the clinician's job. So I am just skipping this because this is the radiation therapy is one of the aspect in the treating the cancer. Next. Next is the major cancer of women. Next. Okay. So there are 5 types of cancer which is most prevalent in the in women. One is breast cancer. Breast cancer, there are mainly two types of cancer. One is arising in the lining of the cells. In the right panel, you can see of the ducts. It is more than 85%, and another is lobular or in the glandular tissue, which is about 15%, and it's the most diagnosed cancer among women worldwide. As I told you, that is 25% of the total cancer patient. About 25% is the breast cancer in women. Another one is ovarian cancer. Actually, it is coming into the ovary. It is originated in the ovary, mostly epithelial cells. And it is earlier it was thought from the ovary, but now it is also in the fallopian epithelial cells in the fallopian tube. Also, it's coming in from that, originate from that. It is actually 5th in cancer death among women, and it is mostly aged people are having this ovarian cancer, but there are some reports also in the young age also. Next. There is also cervical cancer. There are 2 types. One is HPV, human papillomavirus infected cervical cancer, and also another is HPV independent. So this is actually most successful in the treatment. So there is vaccine in the cervical cancer available. And endometrial cancer is another deadly cancer, which is uterine wall. This is endometrium. That is cancer is causing there, and very not so very frequent. That is vaginal and valvular cancer. It is also occurring in women. Next. So the different stages of ovarian cancer, you can see there are mostly 4 stages, but this tumor is like dynamic. So you cannot distinguish from one to another. It is the dynamic stages, and also it is in some cases this is very aggressive, and in some cases it is not so aggressive. So depending upon the different conditions. So in some cases it is growing very fast. In some cases it is not so fast. So there are stages like in the stage 1, when there is a occurrence is found in one or both ovaries. And in the next stage, when the cancerous cells have spread from the ovaries to other parts of the pelvis, such as fallopian tubes, etc. And the next stage is when cancerous cells have spread outside the pelvis. And the last stage is metastasis. The cancer has spread beyond the abdomen and reach the lungs or the spleen. So it can reach up to the lung, and this is the highly metastasized. Next. So there are some symptoms like in ovarian cancer, like pelvic pain, abdominal bloating, fluid collections, back pain. But in sometimes, as I heard from the clinical collaborator, that women does not understand that about this one, and when they know it, it is in the late stage or something. So the symptom is sometimes not very clear. So next. So these are the how you can detect the ovarian cancer by transvaginal ultrasound, TVUS, CT scan. I have already told CA125 marker. It is a protein is coming into the blood, high amount. So, but sometimes it is confusing because benign tumor also giving CA125 sometimes. So, so these are the different strategies by which we can detect, the clinicians can detect the ovarian cancer. Next. So there are different therapies, chemo, hormonal, targeted therapy, immunotherapy. So I have already mentioned. Next one. Okay, now this is the challenge. So here the researchers interest and something. So when I speak about the clinical problems, so the problem is like a clinician's job is very much defined. The patient goes to them, and as a researcher, we have some sort of issues where we can give some sort of our research input so that something can happen in future. For example, the drug development program. So drug development program is another big program. I will come into that. Another one is the how it is not working, how it is working. That is also another area where the researchers are giving their input. Sometimes I am just telling you that the main problem in the ovarian cancer is what I understand is the detection. It is coming into the late detection. There is a not very good early detection marker, and another big problem is the high recurrence of and resistance. So high recurrence and resistance means the treatment is done, then after some times it is relapsing. So why? So because there are now it is very much clear that how it is recurring. So there are another story of the stem cell chems and chemoresistance chem. Some cells are resistant to the drug. So this how this resistance is coming. So there are some drug the pump system which is activated. So they efflux the pump the drugs. Sometimes it is some cells are converted into the stem cells. So what is stem cells? So these cells actually from the normal tumor cells, it becomes tumor stem cells, so that they have the power of some sort of immortality. The drugs cannot attack to them. What? How? Because they are capable of effluxing out the drug, or they cannot they are protected somehow. There is modified, adapted. So these are the things are coming out. This is the concept. See, tumor cells are always trying to survive, right? So if you are giving drugs, they know how to protect themselves. That is the modification, that is the adaptations. So these type of things are coming out, and now new concept is, since the cancer is multifaceted, the many causes are there. So the concept is not a single drug is maybe capable of killing the cancer. There is a concept of multiple or combinatorial therapy. So this recurrence or resistance is coming because some of the cells are out of say millions of cells. Some cells are modified to stem cells. They cannot be killed by the drug. They are like sleeping. When the favorable conditions come, they again form the tumor and the relapse occurs. So this is one area of research where how we can change this or modify this from stemness, how stemness is occurring, how stem cells can be killed. This type of research is important and going on, and several work has been done. Another important is tumor heterogeneity. So there is a concept of tumor heterogeneity, means not even each individual is different. First of all, each individual is different, genetic makeup is different. Even in a tumor, each tumor cells in a tumor, say for example 10 million cells are there. So each cell is different. So the concept is it is too much of heterogeneity is there, and from one tumor type to another tumor type there is a different. So one drug cannot be given to all types of this type of cancer. There are subtypes. So the problem is the heterogeneity makes that is the call that the one size fits for all treatment approach is ineffective. So this is another important aspect. And another is immunotherapy limitations. So immunotherapy is coming a big way now. So you all know. So immunotherapy is given now, but very, very costly, because of especially the CAR-T is very costly. But even in immunotherapy, some cells like in solid tumor, immunotherapy is, especially the CAR-T, is not successful yet. So what is the limitations? Why it is not successful? So that is another area of research. There is a drug delivery and toxicity. That is why, so there is, always there is a toxicity. Everybody knows that chemotherapy has a lot of toxicity issues. So this is another area how to overcome. We have done some work. I'll show you later on how this toxicity can be reduced. There are limited biomarkers. So biomarkers can also be increased. So this is the research is going on how to get more and more biomarker, which is very specific. Next slide, please. So this is the research and development where we can. So next slide. So this is actually when I am working, and many times I am being asked in my family or school friends that you are doing so much of work. What do you have? Any drug you have identified? Have got any drug so far? Then I tell the drug development story. So any drug development needs at least 15 to 25 years from the beginning to the final, if at all it is successful. Okay. So the first stage is target identification, lead discovery, lead optimization, and screening, preclinical lead optimization. So it takes 7 to 10 years to get some molecules. So for any drugs, like in a cancer drugs, if you want to chemotherapeutic drug, if you want to develop, so first you have to synthesize first you have to identify any lead so that we can attack that lead so that the tumor cells is blocked, tumor is killed. So that lead identification. Then you have to synthesize some molecules, chemical synthesis, some molecules, and then those molecules you have to charge with this to block that lead. Then tumor cells will be dying. Then after that, it takes so many years. After that, it is a testing first in mouse, then in dog, then in monkey. After that, it will go to the human trial, phase 1, phase 2, phase 3. Then it will go to the approval. It takes another. So it is minimum 15 to 25 years. So this is the scenario, but it is an ongoing process. Several pharma companies, several organizations are in the process of drug development. You can see in the Google that you can see that there are so many molecules are in phase 1 trial, phase 2 trial, phase 3 trial. So even after phase 3 trial, some drugs cannot come into the market because of the toxicity or different other issues. So drug development story is a very, very detailed thing, and lot of time is required. So I am just giving you some sort of idea about these drug development activities. Next. Okay, this is another thing I would like to let you know, because where we are working, which in the laboratory we are working in taking what? Okay, so we have cells. So every organ is made up of cells. Tumor is also made up of lot of cells. So where the cells are coming from? Either the primary cells from the tumor samples. Clinicians give us tumors from, there is a ethical permissions and everything is there. A lot of permissions are being taken. Cells are coming from the tumor, and that primary cells we can work. It can grow for several days. This is called 2D monolayer. We can see the drug activity there. And then 2D to we can make it to 3D, and from 3D to we can make it to organoid. So from organ tumor samples we can make it to organoid by giving some sort of growth factors or some specific things which we can make again to organoid, and this organoid can be kept in a slide or chip, and this can be used for drug studies, activity studies, etc. And from there, from these tumor samples, we can put it by chopping, we can get the cells and put it into the mouse, and mouse can develop the tumor, and we can study the tumor there for activities of drug, screening of drug, etc., etc. So, these are the different model systems where researchers are interested in or working. So, another thing is like this is a primary cell culture from the tumor we can get. There are some cells which is we can grow for years together. How we can grow? We can make it immortalize. So, these cells can be immortalized by some techniques, and we can grow forever for many years. Okay. So, this is called cell line, okay, and this is primary culture, and we can call it that cell line. So, these are the different aspects, different systems where the researchers are working for different types of mechanism study, drug development, screening, etc., and etc. Next. Okay. So, this is very important. So, now everybody knows that immunotherapy is coming in a big way. So, what is how immunotherapy works? This is very simply it has been mentioned. So, in normal condition, tumor cells, this is the red one is tumor, and this is actually, and the blue one is the inactive in a T cell. T cell means it is T lymphocyte. Actually, it produces some sort of molecule to kill the tumor cells. Okay. So, normal condition, normally when there is no problem, so normally it kills the tumor cells, but sometimes tumor actually producing PD-L1, so that it can it cannot actually it binds to the PD-1 and it is blocking the tumor cell so T cell function, it is inactivated. So, now the tumor cell is live, it is T cell is inactivated. So, PD-L1, PD-1 activity is maintained. So, now what people are doing, they have created some sort of antibody, so that this PD-L1, PD-1 this connection is lost, so that T cell becomes active and it can kill the tumor cells. So, normal tumor cells in my microenvironment, T cell is inactivated because of the tumor cells are trying to survive. Because of it trying to survive, it destroys the immunity. Now, the people have created some sort of antibody or checkpoint inhibitor, it is called checkpoint inhibitors, so that it can block that pathway and tumor cells T cell become active again and it can kill. So, this is the basically checkpoint inhibitor, and it is actually functioning in good ways, and the treatment is going on here. Another important aspect is CAR T cell therapy. This is a cell therapy. This is actually is a maybe future for immunotherapy. It is working more or less well in the blood cancer, liquid cancer, but in solid cancer it is success rate is not that high. So, how it works? So, in the from the patient sample, it takes the cells, T cells. From the patient sample, it takes they take the T cells and they engineer the T cells outside, means laboratory, and then make it a CAR T, and so that it is engineered T cells, so that it can again infuse into the human being and it can kill the tumor cells. So, this is called engineered T cells or adaptive cells or CAR T cells, so that it can modified outside and then again infused into the patient and it kills the cancer cells. So, these two things are this is actually this is actually in the process of in the clinics, but it is coming. It is not very in not all cancers CAR T cell is now successful, but in the especially the solid cancer, solid tumor, it is not yet very much successful, but many work is going on, including our institute. Many work is going on, in many places work is going on. So, hopefully it will be in coming in future. Next. So another concept is like again dual therapy. So here the immunotherapy and chemotherapy. So it is a concept is like some monoclonal antibody tagged with the chemo drugs, so that it can increase the success rate. So here this is called antibody. Another name is antibody drug conjugate. So success rate is high. So you can see the left panel is only the this is the chemotherapy, and another one is chemo plus antibody plus. So it is called, I am just telling you, adjuvant. This is called pembrolizumab, an anti-PD-1 monoclonal antibody, and another one is monoclonal antibody plus immunotherapy. So success rate is very high. So 64% and 51%. Next. So another thing is like this is another concept is coming, that is that combination therapy. I already told. This is a very recent paper published in Cell, the Hallmarks of Cancer by Hanahan. They have this is concept is very old. They have shown that this concept is coming up. So anti-angiogenic therapy as well as PARP inhibitor in combination they are giving, so that it is giving the more success rate. Because the thing is like the concept is like when there is a chemo drug, there is a adaptive feature is increasing, because the tumor cells are are trying to survive, and there is a lot of adaptations. So one drug may not be useful, so that it can from another angle it can be another hallmark is if attacked, then there is a combination therapy could be more successful. Next one. So this is another concept. It is not, although this is the concept is good, but clinically there is not successful so far, not very highly successful. But the concept is like the drug is like there is some sort of photosensitive drug, so it can be incorporated into the cell, and there is some beam of light. It can excite the drug, and it can release the reactive oxygen species. It can activate the ROS level, and it can kill the tumor cells. So this is the photodynamic type of therapy. It is mostly it is not yet maybe not yet coming into the clinically viable drugs, but it is in the concept level. Next. Next is the this is another thing is like targeted delivery. So some tumor cells producing a lot of sort of ligand in the surface, and when you add the drugs with this some nanoparticles with this some sort of attachment where this ligand can bind, so it can only bind to the tumor cells, not the normal cells. So this is little bit of targeted. So the problem of chemo, this is the non-targeted, mostly the normal cells are also dying, but some tumor cells like we have shown that ovarian cancer cells produce a lot of lectins. So so if there is some nanoparticles which can have the lectin binding ligand, so it can bind to that cancer only, not the normal cells. So this type of concept is already there, and some success is also there. Next. Okay, now now this is the next generation. It is a precision medicine or personalized medicine concept. So since every single individual is different, so there is a possibility of their genetic makeup is different. Lot of mutations one person might have, and that another person that they may not have this mutation. So the thing is like presented the treatment is like we are giving the treatment to everyone. If you go to see the lower panel, everyone, and the success rate is somewhere it is success, somewhere there is no successful, it is not successful. But future, if you screen the population, beforehand, before giving the drug, and if you give the proper drug to the proper person, so everyone will be getting the result properly. So this is called the precision medicine or personalized medicine, which is coming in the in near future, or it is already in the concept. Next. Okay, so timeline of major treatment approval for the ovarian cancer. You can see there are already there surgery, chemotherapy since some 1980 onward. So major breakthrough came when 2010 or something, that anti-angiogenic bevacizumab came, it is one, and also the PARP inhibitor. So what is PARP inhibitor? PARP inhibitor is like, it is especially the BRCA1, BRCA2 mutant in people, they cannot, this homologous recombination, the DNA recombination, they cannot make the DNA recombination. So in those BRCA mutant people can be given the PARP inhibitor, so that specifically these cells are dying, so they cannot make the recombination. So these are coming into the 2010 onwards, and now 2020 onwards, see, immunotherapy is coming in the big way, and now the concept is 2030 onwards, it is the combination therapy and precision medicine. So next slide. Okay, so now I am coming to the some aspects of our, so these are the all background information. So some of the important aspects where we are working. So this is so in last 1 decade or 1.5 decades, there is a concept of tumor cells are metabolically modified. Okay. So why this metabolically modified? So what is metabolism? So normally the cells are having the normal metabolic functions, but when it is a tumor cells, their metabolic functions are derailed, deranged, totally reprogrammed. Okay. So this is actually the tumor, normal tumor, where you can see the tumor inside in the core, and there are so tumor cells. There are a lot of other cells in the tumor microenvironment. Okay. Always there is a dynamic interactions between the tumor cells and the other cells in the tumor microenvironment. So this is the dynamic interactions, and I can and there is a always the tumor cells try to survive in such a so that by taking the cues from the other cells in the tumor microenvironments, by taking the signaling help from the other cells or in the tumor microenvironment. So tumor cells are metabolically totally reprogrammed. So this is the concept. The next one. Okay, so our interest is, see, this is the tumor cells and this is the microenvironment. Now there is always a stress factor, quote unquote stress factor. I am telling you stress because tumor cells are there, our immune system trying to kill those cells. There are drugs we are applying that trying to kill the tumor cells. Tumor cells are growing like anything. Normal cells, from normal cells, tumor cells growing like anything. They need lot of nutrients. They need glucose. They need glutamine. They need growth factors. But as they are growing like anything, they are all, sometimes they are not getting as much they require. Okay. So this is the condition, but tumor cells try to survive and they try to grow. So what are the different things are happening inside the tumor cells metabolism? So you can say this is the stress factors, and we can expect the adaptive response from the tumor cells. These are all important in terms of therapeutics. I can tell you why. So stress, there are stress like nutrient deprivation, there is stress like hypoxia, means low oxygen. There is stress like metabolic stress, immune surveillance, growth factor depletion. So these are the stress, and they want to survive. Adaptive features, what are the adaptive features? Metabolic and bioenergetic adaptation, they want to adapt. Signaling alterations, structural modifications, extracellular matrix remodeling, and oncogenic activation. So these are the different things happening in the tumor cells, which is in dynamic condition, always happening. Tumor cell trying to survive, tumor cells trying to adopt, tumor cells adapt. So these are the different types of things happening in the microenvironment. Now our basic question is, what are the different metabolic functions that are changed, and whether we can attack tumor cells by modifying that, giving the tumor inhibitors of these metabolic functions or metabolic modulations. So that we can kill. And in last, you can trust me, in last 1.5 decades, lot of things are going on, and many metabolic modulator inhibitors are there in different stages of clinical trial. I can tell you can see it is now available always in the Google. So you can see how many metabolic modulators or inhibitors are in clinical trials. It may come in next 5-10 years some drugs. So these are the different things which is happening. So now our question is, how we can check it, how we can solve it. Now next slide. Okay, this is very basic things that is happening. I think this is a mixed gathering, so I am not going into the details, but functionally the tumor cells are normal cells. Our normal cells are in presence of oxygen. Our cells go to the TCA cycle. This is oxygen, in presence of oxygen, and we are getting the ATP for energy. And in absence of oxygen, our cells go to the non-mitochondrial glucose pyruvate cycle. Anaerobic glycolysis in absence of oxygen. But tumor cells, even in presence of oxygen, they prefer going through the glycolysis. Don't go for mitochondrial respiration for the ATP generation. This is a major change in the tumor cells, So this is the one major change, but this is the beginning. It was the beginning long ago. Now we can show hundreds of changes are occurring, in the tumor cells are having these changes. Next slide. So these are the different changes, parameters. You can skip next one. Next one. Okay. Our idea was, since tumors are growing, tumors are growing from 1-2 cells to millions of cells, and there is a blood circulation. Always, so there is blood circulation has to be there in tumor cells so that it can get the nutrient and energy. Now, see, the tumor surface cells are getting more prone to blood circulation because they are nearer to the blood capillaries compared to the tumor core, where less blood is going and less nutrient is going. So our interest was to see what is happening in the tumor cells when the nutrients are deprived, nutrients are not giving, because it is the normal condition of the cancer. We are taking the nutrients, the normal cells are getting everywhere, no problem. When there is a tumor, big tumor, the tumor also needs lot of nutrients. They are like Rakhosh, they are taking so much of nutrients, and we are depleting. So now the question is, when there is a deprivation of nutrients, there is a less nutrients, what is happening in the tumor cells? Next slide. Okay, so before going that, we worked with the glutamine. Glutamine and glucose. This is two important things that every cancer cells need. So glutamine enters into the cells by glutamate, and glutamate is formed by the glutaminase enzymes from glutamine to glutamate, and glutamate is entering, and it lot of pathway it goes, giving the glutathione synthesis, amino acid synthesis, and a lot of things, alpha ketoglutarate to TCA cycle. So the basic thing is, don't be confused with all these things. The thing is, we are depriving the tumor cells with nutrients. We don't give the tumor cells nutrients, and then want to check what is happening, whether the cells are behaving normally or dying, or what is happening. You go to the next. Okay. The concept was, if you start the glutamine, so the concept is that blood vessels, nearer to the blood vessels, the glutamine is, the nutrient is available, then the core there will be less nutrient. So what are the effects of glutamine starvation on cancer cell metabolism and growth? And tumor core contains slow growing cells and stem like cancer cells. Our question is there any connection between the glutamine starvation and stemness? And if we block the glutaminase, then whether we can get some sort of good thing or not. So next slide. So if I am not showing the data because it is irrelevant here. So I am showing only one data, that you can see that this is the normal cells growing in the normal medium, cells growing into the glutamine starved condition. So when you deplete the glutamine, this is the picture of mitochondria in the cell. Okay, cellular mitochondria, where the energy is produced, ATP is produced. You can see clearly that when there is a starvation of glutamine, mitochondrial length is shortened. Mitochondria is shortened, and we showed that shortened mitochondria goes to the nuclear periphery, and ultimately there is a induction of this different stem cell marker. So how it is happening, how everything we tried to show, this is a 4-5 years work, so I am telling you in 4 minutes. So the next slide, what we showed here, that when you starve the glutamine, what is happening, mitochondrial fragmentation occurring, and mitochondria goes to the nucleus, and different genes, that is stemness genes, are produced, and normal cells in the core becomes stem cells. So stem cells have the property of resistance, chemoresistance, therapy resistance. So it is possible that when there is a nutrient deprivation in the tumor, some cells becoming stem cells, which when the favorable conditions come, they become cancer again. So this is the concept of the relapse, recurrence of the cancer. So our idea was, if we block the tumor cells by glutaminase inhibitor, the tumor growth will be reduced, no problem. But some cells will become stem and chemoresistance. So we can block the cells by glutaminase inhibitor. At the same time, we can block the DRP1, which is actually chopping the tumor mitochondria. So dual inhibitor of glutaminase and the DRP1 help by blocking the cells as well as preventing it to become stem cells. So dual inhibitors can help, like this is the dual inhibitors, chemically is available. So we treated the cells and also in the animals, and we showed that it is happening. Next slide. So it is actually, you can see that tumors are regressed upon dual treatment. Also we have shown that the stem cells production also reduced. This was our one data. So there are plenty of other data, similar type of things. I am just, next slide. Okay, so this is actually some are in clinical trial, glutaminase inhibitor, not ours, other persons. But we can predict that this glutaminase inhibitor may work, but some cells become resistant. So dual inhibitors might be helpful. Next. So, another thing is metabolites, the cellular metabolism is going on. In the tumor cells, lot of metabolites, new metabolites are accumulated, they are helping the tumor cells to grow. Similar metabolites, one metabolites we identified, next slide. So, these lysophosphatidic acid metabolites are it is a lipid lipophosphatidic acid oncometabolites. So, these metabolites is accumulated in the tumor cells, in the blood the vicinity, tumor microenvironment coming into the blood, which is actually not present in the normal cells. Only tumor cells produce. Why they are producing? Because they act as a signaling molecule for the tumor cells to grow. So, tumor cells producing some metabolites, which in turn helps them to grow more. So, we are there, tumor cells, we are there, we need more. So, they are producing something, and this is called autocrine signaling. So, they have their receptor on their own cells. So, they are produced in much more amount. At the same time, they are helping the producing unsaturated fatty acid, which is required by the tumor cells. So, normal cells they have only phosphatidic acid, but tumor cells having the lysophosphatidic acid, which is absent in the normal cells almost. Next. So, we have shown that this lysophosphatidic acid through signaling, it helps in the tumor progression. So, next. The another aspect also, in earlier slide, go to the earlier slide. Another aspect also is a similar thing. We showed that lactate produced by the tumor cells are going to the nucleus, and their epigenetic modification is going on and cell proliferation is going on. Next. The dual inhibitors, another example. Next slide. I am not going into the more and more examples, but there are so many examples in our laboratory. Dual inhibitors, actually inhibitors for the hexokinase and inhibitors of the lactate transporter, they are, when they are jointly given, then the tumor cell progression is reduced. Another example. Next. So, another example here, that the CPT1, which is otherwise helping the fatty acid oxidation, and tumor cells, the CPT1 production is very high, and it is actually producing more ATP for the tumor cells, and it is growing. And when we block this CPT1, tumor cells progress is reduced. Next slide. Okay. So, this is another thing we did. So, I am giving some of the examples of our work. So, this is actually PARP inhibitor is a blockbuster drug. So, olaparib, rucaparib, these are actually binding to the PARP, and they specifically BRCA mutant or homologous recombinant proficient negative individuals, they are having this type of function, and they are actually selected first genetically, then giving the PARP inhibitor, and then treatment is fantastic, very expensive though, but it is a very good drug. But the problem is lot of hematological toxicity in the PARP inhibitor treatment. So, women are suffering from the low hemoglobin and other toxicities as well. So, the question is, our question was whether or how PARP inhibitor, this is actually purely clinical thing. So, you worked with Dr. Ashima Mukhopadhyay from the Golgo TRG, and it is a part of the project, and it is unpublished data. So, we showed that they showed that instead of giving regular doses, so it is an intermittent dose can be given to some subpopulation of the tumor the patient, whether then it can be reduce the dose and it can reduce the toxicity. Next slide. So, this is one example, like see in the left panel. So, if the dose is given in the 0 day or day 1, up to 7 days the activity remains, up to 3 days activity this is PARP inhibition is there. So it can be given except of daily dose, bi-weekly dose can be given and toxicity can be reduced. And here patient number 11, it cannot be given. So if we screen the patient by means of this testing, the some patient can be given limited dose of or intermittent dose, so that the toxicity can be reduced. In this project also we are having the preparation or synthesis of the PARP inhibitor. The patent is over, so we can get PARP inhibitor in low cost for the Indian. So it is another social aspect. So these two aspects are going on. Next. Okay, so I am at the end of the almost end of the talk. So my question is, my submission is, lot of researches are going on here and there in many institutions, and so what is the outcome of this research? So thing is like the concept of bench to bed concept was there from long ago, but it is not that easy. So we need to so basic researchers need to be translation into the humans, and then patient, then pharma and something like that. So a lot of collaborations, a lot of things are there, which next slide I can tell you one thing, like basically we are doing the discovery science and basic research, investigating the fundamental questions in biology to better understand the cancer formation, progression and the potent interventions, but we have some limitations. So then clinical the clinicians are there, they are giving the treatment to the patients, and collaborations with the clinicians is necessary because of obvious reason. So our question is clinical question, and the clinicians are seeing the patient every day. So the clinical question is more relevant for the basic researchers when there is a collaboration, and the for the drug development and other things, pharmacological and industrial collaboration is essential. So in many cases, like our institute also, these things are taken care these days. Earlier we had researchers used to work alone, but now these things are taken care, clinicians, pharma giant, pharma company and other things, where the research outcome can go to the society. So with this next slide, I end my talk, and I acknowledge my institute, that is CSIR Indian Institute of Chemical Biology, and this is my present lab member. My past lab members, they are in many places now. My collaborators, this is the funding agencies. Next slide. Okay, so these are some of my lab members. These are present, and they are some past lab members. Next. Thank you very much for your patience. Yes. Yes. Yes, sir.
First of all, it's wonderful, the presentation. It gives us insight into a lot of things which we hardly know about. And
Dr. Dujari, I have a point.
yeah,
Before you thank him.
no, I'm not just finally thank him. Right. I have two very naive questions, right, without understanding the details of it. Cancer to common people, even to us, is very dreadful, because once you have cancer, as if it is the end of the road. But with all these newer technologies that you mentioned, newer approach, do you think one day a time will come when conservatively we can say, if you have cancer, there is a chance, a probability of you surviving the cancer?
Very good question. So with the advancement of science, technology, you can see the mortality rate has been reduced drastically. Now, and the clinicians are here, so they can also tell the treatment of the prostate cancer and cervical cancer, the success rate is very, very high, considering the earlier. And also in some cases, if it is diagnosed early, in many cases, many cancers, the treatment is now available and they survive. So it is increasing. Thus, with the advancement of science, the treatment strategy and survivability all are increasing. But it is a slow growing process. So the hopefully things will, one day it will come when success rate will be very very high, and there will be there should not be any panic in the cancer, like in other diseases like fever or any other common cold. The cancer treatment will be more successful and mortality rate will be less. Any other questions? I am ready to take questions because
लेडीज के कैंसर के लिए कुछ लेमन लेडीज के लिए कोई क्लू है क्या कि हमें प्रिकॉशन में क्या करना चाहिए? कोई एक दो चीज जिसे हम लोग कराएं।
Doctor, you have made an excellent exposition of the development and research done in the field of this disease. You have also explained how this disease is formed and how this is trying to be addressed. But your entire discourse is very clinical and it was as if addressing a medical class. As far as the audience is concerned, this audience is a layman audience. So they wanted to know what leads to cancer, their lifestyle, their approach to life, their food habits, their bringing up the children's, and ultimate formation of a body which will be ideal, where such sort of disease do not make home in the body. That part of your general address, which this audience was interested to learn, because all these things in the technical language which you have explained have hardly been understood by the entire audience. So therefore that part for which they had come here to understand the remedy in the normal way of life, this audience is expecting on the basis of their way of living. That remains unaddressed, doctor.
okay.
Thank you, doctor.
Okay. Dr. Chakraborty, are you addressing this issue? Actually, clinicians, doctor is coming, so I have slides. I skipped that. So can you show my the slide? Or
Okay.
actually,
yeah.
I had the slides for this type of predisposition, risk factor, what not to do, what to do. But the thing is, I just
This
since
audience do not understand
so I
what
think
you have presented.
okay.
I can tell, I can address that, sir.
Yeah, so that's why I kept for the clinician so that they can address this. Thank you.
Yeah. Yeah, one more question from here.
Namaste, good afternoon. Thank you for a lovely presentation. Myself, Dr. Vikas Kumar Agarwal, Senior Surgical Oncologist, Apollo Multi-Speciality Hospital. One question.
Yeah.
This microenvironment change can, as a clinician, we see day in, day out, breast cancer, ovarian cancer, same chemotherapy, immunotherapy, everything being given, cancer stem cells, recurrence or metastasis. So can we do microenvironment study and predict or prevent them from happening? One. Second, in a larger perspective, can we utilize those lysophosphorin or tumor lactate and apply as a cheap or a cost-effective way to screen the larger population and prevent them from cancer? Thank you.
So question is very valid. So there is a always tumor microenvironmental change and stem cell is formed, and as you know, the recurrence. But from the tumor samples, it is very difficult, as of now, it is very difficult to study from the tumor cell fells how the stem cell is formed and like that. But we have to find the way. At present, I cannot say that it is possible, but we can think about it. And second is so the outcome of our result can be, can be screened like lysophosphatidic acid and everything. So we are trying actually. This is not like it is stopped. It is trying, but there are some limitations and something. So CA125, like same thing. CA125, lysophosphamide LPA also being tried, but since this is the exact methodology not yet done, how to screen from the serum or something, but we are trying to find out how it can be by means of some colorimetric assay or some other assays. We can see how it can be done from the patient sample. So we are trying actually. We are trying. Okay. Yeah.
Any more questions? Asha, we can take one or two questions. Yeah, please. Yeah, last. Oh, you need a mic.
No, I can hear.
This will not go up to that level. It's limited. Yeah, sure.
Yeah. See, genetic marker identification, and also now clinics are also now doing. So genetic marker, there are panel of genetic marker in ovarian cancer also. There are panel of genetic markers. They are screening. So it is available, and it can be done more widespread, because so there are a lot of mutations. So in breast cancer, ovarian cancer, the mutations are very frequent, very in BRCA also there are so many mutations. So getting sort of total genomic sequence versus chip based sequence. So that is also going on. Chip based sequence is there, but total genomic sequence, and then it is also coming, because sequencing takes 1 or 2, 3, 4 days or 5 days now. So it is coming in the bigger way, but it is going on actually.
Okay, okay. Excuse me, because Dr. Chakraborty has to really go for her very acute job, critical job. So you can ask the questions to the speaker later on when you have a discussion session where she needs to be. But basically, it is not to introduce Dr. Chakraborty, but in our country we read about something called DRDO and something called Indian Defence Forces. It is DRDO which fuels the advancement and the modernization of defense forces. But the DRDO doesn't fight. Who fights? The Indian Army. So today, after the DRDO of oncology, we'll have the Indian Defense Forces of oncology. So she will be telling us how to fight, what are the factors that we need to consider on. I'm sorry that we are a little late, but you must be knowing about her because she's very well known in the field. Not only that, as mentioned by Dr. Tara Dugar, another unpardonable error I have done. I said Mrs. Tara Dugar. It should be Dr. Tara Dugar. She has won enough accolades, enormous amount of accolades, and at present associated with Tata Memorial Center. But she has traveled the world getting training, getting hands-on experiments, hands-on experience on the different aspects of oncology. So I will invite Dr. Chakraborty to please take the stage, and we can have, say, 15-20 minutes discussion according to the schedule given to me. Yeah, sure.
Thank you so much for the kind introduction. मैं बहुत आभारी हूँ और मेरा बहुत सौभाग्य है कि आज मैं आप लोगों के बीच में हूँ. मैं am Bengali, but because the introduction was started in Hindi, and that is a real nice thing to see, because wherever I have traveled all over the world, सारे if it is France, Italy, leave alone UK, सारे अपने language में बात करते हैं, you know, like, but we are still studying after 75 years of independence. हम लोग English में ही बोलते हैं. This has a good and bad side of it. So, you know, हम लोग आशा करेंगे कि ये scientific society आज ये message ले कि, you know, हम लोग अपना language को নিজেদের ভাষাটাকে আমরা বলি, ভালোবাসি এবং সেটাকে এগিয়ে নিয়ে যাই. My heartful thanks for Mr. Dr. Mundra for letting me come here, Dr. Sutara to introduce me, and Mr. Bose, who has been coordinating with me throughout. I don't know what's my good luck to, you know, land up here. Unfortunately, I wanted to show some videos, but that is not working, so the luck doesn't look very good. So I'll sit here instead of going on the dais. I will, pardon me, I'll sit here and try to play some videos as I talk along. तो मैं क्या बोलना चाहती हूँ कि मैं कौन हूँ आपके बीच में. It's a doctor. Does it? इसका क्या मतलब है डॉक्टर का? डॉक्टर का मतलब है कि we treat patients, okay? But what am I doing today here? So I'll start with the thing कि who I am. मैं कौन हूँ actually आपके बीच में? तो I'm a gynae oncologist. So what is gynae oncology? Gynae oncology is a new force, a new change in the society, जिसको हम लोग को अपनाना पड़ेगा, उनको उसका क्रेडिट देना पड़ेगा. मतलब क्या है? मीन्स क्या ज़रूरत है ये गायनी ऑन्कोलॉजी का? हमने अभी जो सुना है, डॉक्टर रॉय हैड गिवन हिज excellent talk on the research, and there is so much. I was just thinking that being a researcher, being a table person, it is not easy, you know. He was saying that he has heard from a clinician कि ovarian cancer gets diagnosed late. So the researchers को कोई clue नहीं है, has no clue that ovarian cancers gets diagnosed late. Why so? I will tell you why it gets late, you know. See, the most common clinical symptoms of ovarian cancer, which is the deadliest cancer in the world. Men, women, similar. It happens only in women, obviously. Because what happens is ovary and fallopian tube is only a very, very small thing. Okay, so it's a very small thing. So if I show a woman's body, if you look into the pelvic area, there is one small 3 to 2 centimeter size, 2 ovaries, and the fallopian tubes, 5 to 5 centimeter size. So from that, it travels whole abdomen of the woman. Now you look into a bigger picture on the right side of the screen. The first thing I'll show you, this is the bladder. This is the uterus, and this is the ovaries and fallopian tubes, and there is rectum. And there are 3 exits in the woman body. Posteriorly is the anus, then comes vagina, then the urethra. So what happens is that small tumor that starts in the small part of the ovary is no way going to create any symptoms. So what happens? The tumor keeps accumulating. वो बढ़ता है, हाँ? And then वो सारा जगह में घूमता है पेट का. Okay, we have got some fluid in the abdomen, that is peritoneal fluid. जो जैसे बच्चे माँ के अंदर जब होते हैं तो एक there is something called amniotic fluid जो बेबी को बड़ा करता है. तो पेट के अंदर भी हम लोग का एक fluid होता है. तो ट्यूमर सेल्स को fluid लेके right टू लेफ्ट तक घूमता रहता है और वो डिपॉजिट होता है पेरिटोनियम में, इधर उधर लीवर के पास पूरा घूमता है. देन क्या होता है, जब वो बहुत बड़ा हो जाता है, देन ओनली द पेशेंट स्टार्ट्स फीलिंग समथिंग इज रॉन्ग. You know, that small ovary, now you see how this looks. This is how it looks when you open the abdomen. हम लोग जब ऑपरेशन करते हैं ये हमको दिखता है. Can you make out where is the ovary? Can you make out where is the uterus? Where is the rectum? No, nowhere. Okay, so there comes the importance of a gynae-oncologist who knows the background, what Dr. Sudeep Roy was trying to say. That, you know, that is, he is saying that wherever Dr. Roy was saying, where all the research and everything and all the science and biology. Okay, so we are the one who try to study this and try to bring little bit of this research to the patient side. And now what happens? I'll tell you one more very important thing here. He was discussing about the drugs, how the chemotherapy works. You know, पहले क्या होता था, ये सब cases जाते थे surgical oncologist के पास. We have a doctor with us, surgical oncologist. You know, ये gynae oncologist कहाँ से आ गया? You know, why not the gynecologist? Why not the obstetricians? What is the role of a gynae oncologist? From where this obstetricians, Dhaimas has become a oncosurgeon? You know, so this is the question of the society today. You know, so we have to break this, these wrong notions. Along with the thing that, you know, that cancer is now almost curable. We have to delegate this cancer. We have to allow the gynae-oncologist to spearhead. They have to take the leadership and treat the gynae cancer because we know the woman. I told you that we have to treat the woman in totality, not that small ovary, you know, because a woman is not only her ovary, it's not only the breast, it's the brain, it's the hypothalamic pituitary axis that is connected with the ovary, you know. So even if a woman attains menopause, she doesn't stop to be woman because the estrogen is not produced. Her needs are unique. Her brain is different from a man, you know. So you need a person who is a gynecologist and also as oncosurgeon, so who understands the gynecology, who understands the cancer and who knows how to treat them. So other thing about this ovarian cancer. Now why I don't give chemo? Now if I see a case like this, I will operate. But I'm 100% sure if she goes outside to a surgeon, they will first give chemo. Because you need to study a lot why she should get only surgery and not chemo. So let me explain why. So what happens when there is so much of tumor? There is definite necrosis, there is no blood supply, the tumors and the cells go into a state where they are in a static S phase. So what happens is they are not growing. So when we take out all this tumor, you know, when we take out all this tumor, the abdomen becomes clean. We call it complete cytoreductive surgery. That means CC0. Not even macroscopic, no tumor is left behind after 12 hours of surgery when we come out of the OT. You know, so what happens? What happens then? The microcells who were in the resting phase, they go into the G phase, that is the growth phase. You know, they start growing. Operation doesn't mean, you know, that we have removed and there are no circulating tumor cells. वो तो रहते हैं. But उनका क्या होता है? They go into a phase where, you know, the chemo can work. Because chemo works always better when there is a better blood supply. So as you reduce the tumor burden, the chemo is becoming more and more effective. So we need to do a very good cytoreduction. Now, unfortunately, I wanted to show this this thing, but it's not working. So just give me 1 minute. I will just escape on this gun. I'll tell, sir. कहाँ पे देख रहे थे हम लोग? नहीं, ये PPT नहीं, tells नहीं. Sorry, my videos are. हाँ वीडियो ना हाँ वो नहीं हो रहा है ना Okay, अच्छा liver let me show the liver okay একটু বড় করা যাবে এটা একটু বড় করে দিন হ্যাঁ No, no, sound লাগবে না. Sound লাগবে না. I will talk. So, as I told you that we are now operating on the liver. So, the gynecologist not only operates on the abdomen down below in the pelvis to remove ovaries because the tumor is going everywhere up to the diaphragm. Diaphragm means when we breathe up and down, there is a muscular area that separates the chest and the abdomen. So, you know, in that area also it goes and we have to operate that and take that part out. So, this is, see, the part of the liver has been taken out. Obviously, it is a multidisciplinary thing. We have the surgeons with us for the liver and other things, but slowly and slowly we are also doing these things. So, now let me go to my presentation. Which one is that? Which one the presentation? Okay, sorry. Okay. So that is what I'm trying to tell is that, you know, it is a very difficult job and what is happening is that it is seen that theoretically when we see that when it is seen that when a gynecologist is involved in the treatment of a woman with cancer, look at her progression-free survival rate. It is so much better. Where did it go? Okay. They are doing so much better compared to the other specialties. So whenever a woman is affected with cancer, there should be a gynecologist who should be, you know, the leader of the team. Of course, there will be the other members. There is multidisciplinary. We also have multidisciplinary team meeting. So now the key benefits, all the research have shown that having a specialized gyne-onco care, it has superior survival rates for the woman. Also 50%, they can do the complete debulking surgery because you need to know why you are doing certain thing, why you are not giving the chemo, why you are taking a 12-hour surgery to the patient. It does accurate staging and adherence to the guidelines. He has shown so many things are there. Guidelines and also a high volume matters like any other places. When you do one thing, same thing every day, you know, you naturally become better at it. So we do the same surgery day in, day out. Our pathologists see the same slides day and out. Okay, our medical oncologists treat, you know, there is a DMG, that is disease management group. So they do the same thing. We have the menopause team, we have the fertility team because now the young women are getting affected and here sir was asking that to address why women are getting, you know, cancer. So see, man or woman, the cancer is a genetic disease. That's what I understand. The genetic, we are all made of gene. We are all made of gene. 2013 the tumor TCGA, that is they came out with the gene print of the everybody, whole human genome. Atlas came out. Okay, so what happens is some genes are hereditary, means some genes the children carry from the parents and they again give it to their children, and there are some genes that we have only in us, that is only linked to us. We cannot give that gene to our offsprings. So what happens is the environmental thing and rest of the other things. So if we have seen so many slides of actually the cell and biology. So if you people have, all of us have seen when একটা ছাদ তৈরি হয়, মানে ছাদ ঢালাই এক্স্যাক্টলি ছাদ ঢালাই যখন বাড়িটা হচ্ছে, ধীরে ধীরে বাড়ি হচ্ছে, লোকে জিনিসপত্র নিচ্ছে, বাট সামথিং টু সি ইজ ছাদ ঢালাই মানে কি, মানে হচ্ছে প্রচণ্ড আওয়াজ হচ্ছে, চারিদিকে মোটর চলছে, সিমেন্ট মেশানো হচ্ছে, এ ওপরে উঠছে, এদিকে নামছে, এদিকে ছুটছে, ওদিকে ছুটছে। তো অ্যাকচুয়ালি আওয়ার হোয়াট ইজ হ্যাপেনিং ইন আওয়ার সেল, all the time we are sitting, each cell is like a ঢালাই গোয়িং অন, হ্যাঁ, ছাদ ঢালাই হচ্ছে। তার ফলে হোয়াট ইজ হ্যাপেনিং ইজ দের ইজ লট অফ ডেব্রিক্স অ্যারাউন্ড। নাও there when there is lot of accumulation of debris, suddenly the body may stop working. তো এখানে আমাদের বডির জিনগুলোর কাজ কি হচ্ছে, দ্য রিপেয়ার জিনস অ্যান্ড দ্য প্রোটেকশন জিনস, যেরকম ব্র্যাকা বা মিসম্যাচ রিপেয়ার জিনস, এইগুলোর আসল কাজ হচ্ছে ক্যান্সার প্রোটেকশন। তো এরা কি করে, যখন আমাদের শরীরের সব সেল ফিজিওলজিক্যাল কাজগুলো করছে ছাদ ঢালাইয়ের মতো, ওরা কি করে, ওরা ওগুলোকে পরিষ্কার করে দিচ্ছে। মানে দে আর অ্যাকচুয়ালি দ্য কি বলবো আমরা, মানে সাফাইওয়ালাস অ্যান্ড সাফাইওয়ালিস অফ দ্য সেল। তো ওরা ওগুলোকে নিয়ে যাচ্ছে। তাই আমাদের কোনো জেনেটিক ডেব্রিক্স হচ্ছে না এবং আমাদের জেনেটিক কোনো মিউটেশন হচ্ছে না, সো উই আর প্রোটেক্টেড। আর যদিও বা হচ্ছে একটা দুটো, আমাদের ইমিউনিটি ইজ গুড এনাফ টু রিমুভ দেম। তো দিস ইজ দ্য বেসিক থিং। এখন এই মোমেন্টে যদি একটা টুইক কোথাও হয়ে যায়, যে একটা সেলে মিউটেশন হয়ে গেল, আর এই মিউটেটেড সেলগুলো ধীরে 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 বাড়তে থাকলো, অ্যান্ড দে টেক ওভার দ্য হিউম্যান সেলুলার বায়োলজি, তখন থেকে ক্যান্সারের শুরু। তো এটা সেম উইথ এভরিবডি, এনি ক্যান্সার ইট ইজ সেম। Now, anything specific to what women can do or what girls can do to prevent them from cancer, তাহলে আমাকে বলতে হয় যে যদি কারো ফ্যামিলিতে দেখে কারো ব্রেস্ট ক্যান্সার থাকে বা কারো ওভেরিয়ান ক্যান্সার বা আননোন ক্যান্সার, একজনের ঠাকুমা মারা গেছিল, পেটটা ফুলে গেছিল, আমি জানি না, বাট ইউ টক, you bring your family together, sit together during dinner, খাওয়ার সময় বসে লেটস ডিসকাস, ওই মাসির কি ব্রেস্ট ক্যান্সার হয়েছিল, তখন ওনার বয়স কত ছিল। যত কম বয়সে ক্যান্সার হয়েছে, আমাদের লাইটটা তাড়াতাড়ি জ্বলা উচিত, যে ক্যান্সার ইজ অ্যাকচুয়ালি এ ডিজিজ অফ ওল্ড এজ। তার কারণ এখানে একটা নাডসন্স থিওরি অফ মিউটেশন রয়েছে, মানে আমরা সবাই দুটো করে ভালো জিন নিয়ে জন্মেছি। সেটাই কি হচ্ছে, যে আমাদের সিস্টেমগুলো সব কাজ করছে, যত বয়স বাড়ছে জিনগুলো কিন্তু আমাদের টিভির মতো বা ইসের মতো ধীরে ধীরে খারাপ হবেই, হ্যাঁ। তো একটা খারাপ হলো, তখন আরেকটা কাজ করছে, যখন বয়স হলো তখন দুটোই খারাপ হয়ে গেল, তাহলে ক্যান্সার হলো। কিন্তু হেরিডিটারি মানে কি, দ্যাট পারসন ইজ বর্ন উইথ সামথিং অলরেডি মিউটেটেড। তো ওর কি হবে, ওর একটাই ভালো জিন আছে, তো ওইটাও যদি খারাপ হয়ে যাবে, তো 40 ইয়ার্স এজে, তো ওদের ক্যান্সারটা আগে হওয়ার চান্স অনেক বেশি, হবেই যে তা নয়, কিন্তু হওয়ার চান্সটা বেশি। তো এখানে সবসময় ফ্যামিলিতে একটা অ্যালার্টনেস দরকার, কি হ্যাঁ আমাদের কি কারো ক্যান্সার ছিল, লেটস টক অ্যাবাউট ইট। দ্যাট ইজ ভেরি মাচ রেলেভেন্ট টু ব্রেস্ট অ্যান্ড ওভেরিয়ান ক্যান্সার, বিকজ ইট ইজ কল্ড হেরিডিটারি ব্রেস্ট ওভেরিয়ান ক্যান্সার, যেটা ফ্যামিলিতে চলে, ইটস নট ভেরি আনকমন। ইভেন অ্যাঞ্জেলিনা জোলি, ইউ হ্যাভ হার্ড দ্যাট শি রিমুভড হার ওভারিজ অ্যান্ড হার ব্রেস্ট, ওকে। সো অ্যান্ড দের আর মেনি লাইক দ্যাট। আরেকটা হচ্ছে সার্ভাইকাল ক্যান্সার, যেটায় আমরা কি করে আমাদের বাচ্চা মেয়েদের বা ছেলেদের প্রোটেক্ট করব এই এইচপিভি মানে হচ্ছে হিউম্যান প্যাপিলোমা ভাইরাস রিলেটেড ক্যান্সারগুলো থেকে। আমি মেয়েদেরটাতেই আসছি। এখন একটা জিনিস হচ্ছে যে পৃথিবীতে আমরা যদি মানে ডেভেলপড কান্ট্রিজগুলোতে দেখি, হোয়াট হ্যাপেন্স ইজ দ্যাট 70% কাভারেজ অফ স্ক্রিনিং মানে কি, এইচপিভি through pap smear or through HPV detection, screening. There's a recall system. They will call you. Their GPs will call you. এখানে ওসব কিচ্ছু নেই, হ্যাঁ, nothing. We are only India is 2 to 3% covered for screening. তো আমাদের পুরো দায়িত্ব আমাদের নিজেদের, ঠিক আছে। এদিক ওদিক অর্গানাইজেশনসগুলো করছে, এনজিওরা করছে, বাট উই হ্যাভ রিচড নোওয়্যার where we can give 60-70% স্ক্রিনিং ফর আওয়ার ওমেন। তো এটা তো আমাদের নিজেদের ঠিক থাকতে হবে। তো হাউ ডু উই, এখন ভ্যাকসিন এসে গেছে, অ্যাটলিস্ট একটা ভ্যাকসিনও যদি আমরা বাচ্চা মেয়েদের দিতে পারি, দিস ইজ গুড। আপ টু 45 ইয়ার্স ওমেন ইট ইজ রেকমেন্ডেড তারা নিতে পারে, ইরেসপেক্টিভ অফ দে আর HPV এক্সপোজ, সেক্সুয়ালি অ্যাক্টিভ, or they had children, doesn't matter. So the good vaccine is there. It is excellent, it has very good effective থিং, বাট আমরা এই প্যাপসমিয়ার আর এইচপিভিটাকে যেন ভুলে না যাই। যখন ক্লিনিকে যাই গাইনোকোলজিস্টের কাছে, গাইনোকোলজিস্ট শুড নট লুক অ্যাট ইউর ফেস, লিখে একটা প্রেসক্রিপশন লিখে দিলাম, হ্যাঁ, এটা যেন না হয়। প্লিজ when you go or take somebody to a gynecologist, make sure that he or she should examine you down below, বিকজ আপনার মুখ দেখে কেউ বলতে পারবে না কি আপনার সার্ভিক্সে ক্যান্সার হয়েছে, কি হচ্ছে, কি আপনার ভেজাইনাতে লাইকেন স্ক্লেরোসাস আছে, কি একটা অ্যাবনর্মাল কিছু হচ্ছে। দ্যাট ইজ দ্য জব অফ গাইনোকোলজিস্ট। আই এম টেলিং ইউ ভেরি ব্লান্টলি বিকজ উই গেট প্রেসক্রিপশনস অ্যান্ড ওমেন আর কামিং, ভেজাইনাল ডিসচার্জ হয়ে যাচ্ছে 5 বছর 7 বছর অ্যান্ড কামিং টু আস ইন স্টেজ 4 সার্ভাইকাল ক্যান্সার। নো, কেউ যদি একটু স্পেকুলামটা ঢুকিয়ে তাকে দেখতো, শি উড হ্যাভ বিন সেভ টুডে। তো সেটা ইম্পর্টেন্ট। আরেকটা হচ্ছে বাচ্চাদের জন্য ভ্যাকসিনস যেরকম রয়েছে, গুড হ্যাবিটস, গুড ফুড, আজকে জাঙ্ক ফুড এগুলো তারা বলে লাভ নেই। ইম্পর্টেন্ট জিনিস হচ্ছে হাইজিন, সেক্সুয়াল হাইজিন, ইউজ অফ কন্ডোমস, ইট প্রিভেন্টস অল সর্টস অফ ক্যান্সার ইনক্লুডিং সার্ভাইকাল ক্যান্সার। এন্ডোমেট্রিয়াল ক্যান্সারে যদি আসি, তাহলে এন্ডোমেট্রিয়াম মানে হচ্ছে ইউট্রাসের ক্যান্সার। এগুলো আমি তাড়াতাড়ি করে দিচ্ছি। তো ইউট্রাসের ক্যান্সারে যদি আসি, আমরা দেখতে পাচ্ছি যে দ্য ফ্যাক্টরস, এখন যেটা হচ্ছে আমাদের খুব বসে থাকার লাইফস্টাইল এবং দ্য ওবেসিটি অ্যান্ড এভরিথিং, অ্যান্ড দ্য চিলড্রেন আর ভেরি বিগ ইন সাইজ অ্যাকচুয়ালি। তো সেইগুলো থেকে একটু অ্যাক্টিভিটিস থাকতে হবে, অ্যান্ড women should report any symptoms, any bleeding, anything. Uterine cancer is one of the best cancers because bleeding হলে কেউ বসে থাকে না বাড়িতে, সবাই চলে যাবে। So it is always diagnosed very early, it is treated early, so সেখানে ভয়ের কিছু নেই। And definitely everything can spread everywhere, and this is again a very theoretical thing. আমি এটাকে একটু ভেঙে যদি বলে দিই, তাহলে হচ্ছে যে যখন কেউ একটা সিমটমস নিয়ে আসে, তাকে লোকাল এক্সামিনেশন মানে পার স্পেকুলাম এক্সামিনেশন করতে হবে, পার রেকটাল করতে হবে, তারপরে হচ্ছে একটা ট্রান্সভেজাইনাল আল্ট্রাসাউন্ড দিয়ে দেখতে হবে, দেখে তারপরে উই ক্যান ডু এ বায়োপসি। আর এখানে রয়েছে হাউ দ্য ক্যান্সার, ডিফারেন্ট টাইপস অফ ক্যান্সার্স কী করে হয়, ওটা আমি তাড়াতাড়ি ওটার বলছি না। তো বেসিক্যালি আমরা এখন লেটেস্ট একটা ইউট্রাইন ক্যান্সারে আমাদের সেন্টিনাল লিম্ফ নোড বায়োপসি এসেছে। তো সেন্টিনাল লিম্ফ নোড biopsy is where we put the ICG dye in the cervix. তো what is happening in that is, আগে যেটা হতো প্রচুর লিম্ফ নোডস বার করা হতো, and the woman used to come with big swelling on the legs, big surgery, lymphedema. তো এখন আমরা জাস্ট ফার্স্ট নোড যেটা চলছে না নট হ্যাঁ চলছে না, হ্যাঁ সরি, আমার স্লাইডগুলো একটাও আমার সঙ্গে সাথ দিল না। যাই হোক, তো আমরা যেটা করি যে আমরা সেন্টিনাল নোট করে ফার্স্ট নোটটা বার করি, তাতে দ্য টাইম অফ সার্জারি হ্যাজ কাম ডাউন অ্যান্ড দ্য কমপ্লিকেশনস অফ লিম্ফেডেনেক্টমি হ্যাজ কাম ডাউন, সেটা, অ্যান্ড women can go home the next day with robotic and minimal access surgery. I can show you one small video, এখানে হয়ে যাবে আশা করি। কোনটা, এটা না, এটাতে যদি আমি চাই, এনিথিং ইউ ওয়ান্ট টু আস্ক, মানে ইয়া, ইয়েস, টোব্যাকো, okay, so tobacco is no, tobacco is a cause of cervical cancer. It is associated, when we take history, we always ask a history of personal history. Our women drugs, it is only tobacco usually. We do not really come across the drug part of it, so we don't see in our clinics ki, and I have at least not seen, but tobacco and tamaku, guraku, natmaja, এগুলো ভীষণ কমন, অ্যান্ড ওদের বললেই বলবে আপনি কবে থেকে নিচ্ছেন না, এই তো ডায়াগনোসিস, এখন আর নিচ্ছি না, আমি লাস্ট 35 ইয়ার্স নিয়েছি, এখন আর নিচ্ছি না। তো এই আর কি, তো ওগুলো তো ছাড়তেই হবে, কমপ্লিট নো ফর দ্যাট, but you cannot explain why some chain smoker doesn't have anything and why somebody who never smoked has something, so these are also tumor biology that we have to accept. And alcohol, previously the WHO has said we can take up to 1 to 2 units per week, but now it is a complete no. Any alcohol is harmful and it doesn't cause cancer in a day, but it does cause it does cause cancers. It is cancerous. So I'll just try to show you the sentinel node biopsy. And other thing is, last but not the least, is the patient advocacy. Patient advocacy means we tried the patients who had actually, just give me 1 minute. Actually, I had such a short time to, like prepare to, yeah. So I couldn't really look into those things. Sorry about that. No, that's okay. I'm not looking for this. This is, this I'll show you that how close we are working. When we work on the liver, you can see something pulsating. That is the heart. Can you make out? Something is pulsating. So we work that close to the heart. So the liver, with the tumor, the liver is totally close to the diaphragm, so we have to work very gently and to work like that. I wanted to show the sentinel thing, but I don't know. Recently. So this is what the uterus looks like. একটা লাইট অফ করে দিলে একটু বেটার দেখা যাবে। ফ্রন্টাল লাইটটা একটু So here we have done the sentinel node. This is a uterine cancer. Uterine cancer is usually seen inside uterus. So we are seeing the uterus from through the laparoscope, and you have already removed the nodes, the lymph nodes. Why lymph node is important? The lymphadenectomy shows us the stage, so that you know whether the woman will need chemo or radiation. That is also very important. হ্যাঁ সেন্টিনেলটা আমি একটু দেখাতে চাইছিলাম ইটস ভেরি ইন্টারেস্টিং okay 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 yeah so I'll just I will just say because it is very late, and thank you for giving me the opportunity. So I want to tell something about the advocacy part. Advocacy is very important. Means the women who have who are the survivors or the warriors. We don't call them victim, we call them warriors. We have made a team and they motivate others, and so we need a big team, because it's a very lonely, the cancer treatment and everything is a very lonely path. So we should not make anybody feel left out. So we should help them. And this is just, I thought I will give a curtain raiser for our, speciality. So thank you so much. हाँ अभी it is. Thank you sir.
Now I will request the president of this meeting to make his remarks.
Hello, wonderful audience. The Honorable chairman of this institution, Dr. Vitthal Das Mundra. All others are doctor on the dais except me. I am the only person without a doctor degree. Some are expert in their, they are all expert in their respective fields. It's a great effort on the part of Dr. Mundra to have called for such a wonderful meeting where experts on a very live subject which requires full attention and knowledge has been discussed. Dr. Roy, being a research scholar, having been involved in this exercise for more than 26 years now, has given the imprint of all that which he has done as a research scholar in such a 1 hour plus demonstration. But Dr. Chakravarti has come out with many things practical which needs to be addressed. I want to have even more information for the women in general as to what protection and what health consciousness they should have, so that this endemic increasing disease can be addressed and our women can remain healthy. जननी जन्मभूमि, जननी और जन्मभूमि दोनों ही इस राष्ट्र के लिए जरूरी है. जननी अगर स्वस्थ है, जननी अगर सुंदर है, जननी अगर राधा है, तो हर आदमी मोहन है. इसलिए जननी का स्वास्थ्य, जननी का सौंदर्य, जननी का जीवन बहुत इंपॉर्टेंट है. इसलिए हमारे समाज की हर नारी स्वस्थ हो, सुंदर हो, कैंसर फ्री हो, इसका एक हमें अभियान चलाना है. और वो अभियान मेडिकली टेस्टेड अप्रोच कि भाई ठीक है, कुछ तो जेनेटिक हैं, वो जेनेटिक अगर हैं तो उनके परिवार का है तो शुरू दिन से उसको कैसे एड्रेस किया जाए कि वो आने वाली जनरेशन में प्रिवेंट किया जा सके, या जिनमें चांसेस हैं आने का एक कंपेन चलाया जाए कि वो इन सब चीजों को एड्रेस करें जिसमें उनके अंदर में ना आए. आज कुछ कुरीतियां हमारे समाज में आ रही है. लड़कियां सिगरेट पीने लगी हैं, लड़कियां शराब पीने लगी हैं, लड़कियां फास्ट लाइफ लीड करने लगी हैं, लड़कियां फास्ट फूड खाने लगी हैं. तो ये बहुत कुछ इग्नोरेंस की वजह से हो रहा है. अगर उनको ये समझ में आएगा कि इन सब चीजों का सेवन हमारे समाज के लिए, उनके स्वास्थ्य के लिए, उनके आने वाले बच्चों के लिए, क्योंकि उनके शरीर में अगर कोई रोग आ गया तो उनके बच्चों में भी रोग आ जाएगा, तो एनेडेमिक हो गया, जेनेटिक हो गया. तो इसलिए उनका शरीर स्वस्थ और सुंदर रहे, जिससे आने वाली संतति स्वस्थ और सुंदर रहे. ये कैंपेन चलाना बहुत जरूरी है. इसलिए आज का जो मीटिंग है ये सब में एक अवेयरनेस पैदा करे कि कैंसर एक ऐसा डिजीज है जो समाज को खा जाता है, जो वुमेन के शरीर को खा जाता है. ये हमें प्रिवेंट करना है. तो ये पर्पस अगर सर्व होता है तो आज की मीटिंग बहुत ही बढ़िया है. थैंक यू सो मच.
We have Mr. Rajgopal Shureka to give vote of thanks. But prior to that, you want to say something? No, please, please, right.
मित्रों, आज शिवरात्रि का दिन है और भगवान शंकर की पूजा रात को सब हम करेंगे. तो भगवान शंकर को प्रार्थना करते हुए आप सबने पधार कर हमारा मान बढ़ाया. खास तौर से तुलसियान जी, डॉक्टर रॉय, डॉक्टर चक्रवर्ती और हमारे मित्र डॉक्टर दुआरी का तो हम हर इन सब का हम बहुत बहुत आभार मानते हैं. और राजगोपाल जी कहाँ है भाई? वो बैकग्राउंड में है. लेकिन एक बात क्या है, इसी जैसा कि डॉक्टर दुआरी ने पहले कहा, विज्ञान और समाज कि लोग, उनके बीच के अंदर एक दूरी बनी हुई थी। वे अब डॉक्टर राय क्या कर रहे थे हमको क्या मालूम। और उसके पहले हमारे जो अब ये ग्लास एंड सिरेमिक रिसर्च इंस्टीट्यूट के जो डायरेक्टर है उनका भाषण सुना। इस तरह से बहुत से लोग हैं हमारे समाज के बीच में। हम उनके महत्व को जाने, आंखें, और साथ में भाई उनको भी तो कुछ सहयोग की आवश्यकता है। इट्स द रेस्पॉन्सिबिलिटी ऑफ द सोसाइटी टू सपोर्ट देम। एंड जैसा आज इस कैंसर के मामले में तुलसीराम जी ने जैसा कहा और डॉक्टर चक्रवर्ती ने जैसा हिंट किया था कि एक कैंपेन चलाने की जरूरत है जिससे कि ज्यादा स्क्रीनिंग हो सके और स्वस्थ जीवन के प्रति लोगों को अवेयर कराया जा सकते। तो हम इन वैज्ञानिकों से जैसे जैसे मिलेंगे, हम इनसे बात करेंगे, बहुत सी बातें इस देश को, इस समाज को इंप्रूव करने के लिए आएगी। और एक बात और है मित्रों, हम बहुत सी बातों के अंदर हम उम्मीद करते हैं कि सरकार कर देगी। भाई साहब, सरकार सब कुछ नहीं कर सकती। अपने खेत की जो बाउंड्री होती है, जिसको हम लोग राजस्थानी में बाड़ बोलते हैं, कांटों का बाउंड, सरकार सुरक्षा दे सकती है, सरकार पानी लाके दे सकती है, सरकार फर्टिलाइजर आपको प्रोवाइड कर सकती है, बीज भी दे सकती है, लेकिन खेती तो आपको करनी पड़ेगी भाई। और वो खेती एक अकेला नहीं कर सकता, अच्छी खेती मिलके करनी पड़ती है। और इस अकेली खेती नहीं कर सकते तो समाज के सारे अंगों को मिलकर के जब हम समाज का निर्माण करेंगे तो बहुत सुंदर खेती आएगी। और देखिए, भूलिए मत, ये बहुत परिवर्तनशील युग है। इसके अंदर या तो हम ऊपर जाएंगे या एकदम नीचे जाएंगे। तो इसलिए इस समय हमको साथ के अंदर मिलकर ये काम करना पड़ेगा। मैं अंत में कहूंगा, ओम सहनावतु सहनौ भुनक्तु सह वीर्यं करवावहि तेजस्विनावधीतमस्तु मा विद्विषावहि। ओम शांति शांति शांति। बहुत बहुत धन्यवाद। डॉक्टर साहब, आप कुछ कह रहे थे?
मैं तो सीखने आया हूँ, सीख रहा हूँ।
इधर आओ बंधु। इनके जैसे आगे का जो जमाना है इन जैसे यंग आदमियों के हाथ में है। तो आप और क्या
21 साल को बेटो है।
तो क्या हो, हमारे 60 साल को बेटो है तो क्या हो।
हमारी शादी 16 साल में हुई थी, हमारी 30 में हुई।
बात हरी मर्जी। सो एनीवे, तो आगे का जो अब देखिए, हमारा तो समय आ गया, किनारे के पेड़ हैं, ऐसे जवान आदमी तो ये लोग आगे संभालेंगे, इनको आगे बढ़ाना है। बहुत बहुत धन्यवाद। अब आप भोजन करके पधारिएगा। ओहो, तो वहां पर पीछे छिपी हुई हमको कैसे मालूम?
इनका सुझाव है कि आज इंटरनेशनल चिल्ड्रन कैंसर डे भी है, तो ये चाहती है कि अपने को इसको बहुत अर्ली स्टेज में स्कूलों में, विद्यालयों में इसको हमको कैंपेन चलाना चाहिए।
तो ये बताएं कैसे, क्या तरीका है इसका, फिर आपसे बात करेंगे।
पकड़ा है कि भारतीय विद्या मंदिर से ये प्रारंभ करेंगे।
तो बहुत बढ़िया, बहुत बढ़िया, बहुत कृपा। तो रावत जी, आप इनसे क्या क्या कार्यक्रम करना चाहिए वो ले लीजिए। तो अब भोजन करने के लिए पधारे हैं।